Free radical formation. Here, homolytic cleavage of covalent bond, in which a normal molecule fragments in two, each fragment retaining one of the paired electrons. Now, when you are going to consider homolytic cleavage, when there is the formation of the molecular structure, there will be the sharing of the electrons between the two different atoms or the molecules within the particular compound. For example, if you are going to take the carbohydrates itself, there the carbon, hydrogen and oxygen, these three different elements are going to get attached by the bonding nature. Because of this, what will happen during this bonding nature, there will be the sharing of the electron and it is going to become a particular complex or a particular biomolecular structure. When these bondings are going to cleave, during this cleaving of the bonding, there will be the separation of the electron pair where these bondings are get already attached. Because of this, one of the paired electron is get retained in one of the molecule and the other electron is going to be given to the other molecule. If this is going to happen, during this time, these molecules or the atoms are going to become the free radicals. Here again, homolytic cleavage occurs less commonly in biological systems as it requires high energy input from ultraviolet light, heat or ionizing radiations. So here within the biological systems, this kind of the cleavages will take place very little. So here you can see it's a depiction of what are the free radicals, where the free radicals are going to act as robbers, which are the deficient in energy, and also they are going to damage the cells. So those are caused by these factors on where the free radicals are going to show the effect on the cell membrane and internal organs and the nucleus also. Now coming to the next slide, free radicals, how they are formed, the continuation. The chemicals cortisone and catecholamines created by metal stress can create free radicals. So here if you are going to take these chemicals, they can cause the metal stress and can create the free radicals. Body's free radical molecules are a natural byproduct of cell metabolism. As we have already discussed in the first slide during the definition and description, that I have spoken that the metabolism is where the byproduct free radical has been observed. So even in the alcohol consumption, it can be the any amount of the production of free radicals within the body can takes place. And again, polyunsaturated fat like which is found in vegetables, that is vegetable oils, is easily oxidized in the body and can create free radicals. Now here it has to be substituted with polyunsaturated fats with monosaturated fat. So here when you are going to consider this polyunsaturated fat is dangerous, that is it is going to create the free radicals within the body. Now you can see the causes of the free radicals formation or the sources. Now coming to the free radical source, here there can be two sources of the free radical formation, that is the internal source of free radical generation and the external source of the free radical generation. Now internal source, as we have discussed already, that the free radicals are the byproducts of the metabolism, as the metabolism is the internal process. So that cell metabolism is going to stand as the internal source of the free radical generation. Now under this, the enzymatic reactions and the respiratory chains which takes place in the internal organelles of the cell, and the phagocytosis, it is also a internal process. Prostaglandin synthesis, even that cytochrome P450 system is also an internal process of the organelle, that is the mitochondria. And xanthine oxidase, peroxisomes, inflammation, and arachidonate pathways, exercise that we do every day, even it is going to cause the metabolic difference, and it can cause the metabolic byproduct as the free radical, that is the ischemia, reperfusion injury, or the internal sources for the generation of the free radical. Now further, when you are going to consider the external sources are non-enzymatic reactions of oxygen with organic compounds, any organic compounds which you are going to get into contact, that is you can consume also, so because of that it can happen. And also it can be the ionizing radiations like UV, X-ray, gamma, alpha, like this, and the use of the cigarette, environmental pollutants, certain drugs what we consume for our health, pesticides, industrial solvents, ozone, X-rays, etc. Now here you have seen a bunch of the reasons which are majorly causing the free radical formation within the human beings, that is during the metabolic processes as the internal source and as the non-metabolic process. It will also be a little kind of the metabolic process only, but the effect or the factor will be the external source, not the internal source. Now when you are going to consider, here I have mentioned certain processes out of these internal and external, but I have taken majorly of the internal processes, that is the xanthine oxidase. Xanthine oxidase is the kind of the xanthine oxidoreductase, where it is a kind of the enzyme which generates reactive oxygen species. These enzymes catalyzes the oxidation of hypoxanthine to xanthine and can further catalyze the oxidation of xanthine to uric acid. So it is a catabolic process where they are going to catalyze, where they are going to break down the xanthine to uric acid. Or further, these enzymes play an important role in the catabolism of purines in some species, including humans. Now here during this catabolism, there will be the, you can see, there will be the oxidase action on the uric acids, where there will be the release of CO3 minus, and here there will be the formation of uric acid, but there will be the release of H2O plus O2 minus, and here you can see peroxynitrate NO plus O2, where there will be the effect on the cytochrome C reductase. Here there will be the effect on formation of the NO plus O2. So here out of this whole reaction, you have come to know that there will be the generation of the free radicals as the byproduct within these metabolic cycles. Now when you are going to consider the same in the peroxisomes, so here in the peroxisomes you can see this metabolic activity happening, where this there will be the oxidases within this particular peroxisomes. These are the organelles which will be present within this cells where they are going to do a catabolic process where they are going to generate the free radicals of hydrogen peroxide, where these hydrogen peroxide get influenced by the metal ions, where these are going to generate the hydroxyls, where these hydroxyls are going to have the difference in their valence of electron, and they are going to affect the cell membrane, peroxisomal membrane damage. Now in the same manner you can see here the same has been elaborated, where it can be the O2 minus or it can be the H2O plus O2 oxygen. So here this is how the metabolic activities are going to become a source to generate the free radicals. Now in the same manner, if you are going to consider in this metabolic activity like ischemia, reperfusion injury, IRI or oxygenation injury, is the tissue damage caused when blood supply returns to tissue, that is reperfusion, after a period of ischemia or lack of oxygen, that is anoxia or hypoxia. Now here, as per the description, that the first stage is the ischemia, that is the lack of oxygen, or the anoxia or hypoxia. Here what will happen, if you are going to see this diagram, this diagram shows that there is the reduced flow of the oxygen which will be observed. Here this is caused because of the some depositions which is going to happen within the blood vessels. Due to that, what will happen, the blood RBCs which are carrying the oxygens, they cannot deliver the oxygen, and these cells which are present at that location cannot absorb the oxygen. During this time what will happen, there will be the reduced amount of the oxygen, where these cells will be having the function of taking the oxygen from the hemoglobin or the RBCs, where these are going to fail to absorb the oxygen. Because of that, what will happen, there will be the reduced oxygen supplied to the cells, where anaerobic glycolysis is going to increase, and this leads to the formation of the lactate, and which is going to reduce the formation of the pH. Due to the lack in the pH, the activity of the mitochondria is going to reduce. Now here on the other side, the same reduced oxygen condition of the cell leads to the ATP hydrolysis, which will lead to the efflux of the hydrogen atoms and influx of the sodium, where it is also going to lead to the efflux of the sodium and influx of the calcium. This calcium influences the MPTP opening of the mitochondria. It is a kind of catabolic process where that is not needed here, as we are discussing the free radicals formation. So we are making it to very short and very understandable. Now here, after this process, if these cells are going to get the oxygen, that is the oxygen will be restored. This ischemia past cells are going to get the oxygen. This oxygen will be restored, and this is going to be, because of the restoration of the oxygen, the formation of the lactic acid will take place. This increases the pH. The pH is going to induct the MPTP opening, where it is going to give the catabolic process in the mitochondria, where the mitochondria releases the reactive oxygen species. Now here, the restored oxygen again is going to have the oxygen which is going to combine to form the reactive oxygen species. The same reactive oxygen species again is going to induct the signal to MPTP opening at the mitochondria. Now the same ROS, that is the reactive oxygen species, is going to reduce or decrease the influx of the sodium and efflux of the hydrogen, and also it is going to reduce or stop the efflux of the calcium and influx of the sodium. So this is how the reperfusion is going to generate this particular form of the free radicals within this particular phenomena of ischemia reperfusion injury. So this is regarding how the metabolic activities of the cells during the imbalance in the blood flow with the oxygen or not with the oxygen is going to lead to the generation of the ROS, reactive oxygen species. These are the metabolic functions where we are seeing the formation of the free radicals, where these are going to become the source for the free radical formation within the cells. Now, when you are going to consider the internal sources, these internal sources are going to generate the free radical either by accidentally or deliberately. Now we are going to discuss the accidental kind of the free radical generation. Now here that is going to happen when there is the interface of the bacterium and the activated phagocyte. When you are going to consider interface of the bacterium is nothing but when the bacterium is going to infect the body, it is going to be considered as the pathogen, and that pathogen is going to be get engulfed, that is by the macrophages, in the form of the phagocytosis process. Now here when you are going to consider this particular process is going to be get activated because of the influence of the bacterium. These bacterium are going to get influenced on the body accidentally, suddenly. Because of that, this particular process is going to takes place. During this process, what will happen? The pathogen is going to get engulfed, and that will be moved into the cell where a phagosome will be formed. Here I have made two diagrams. This is the section kind of the diagram which is above. The below is the complete diagram. Here you can see pathogens engulfing by the macrophage, and it is going to be moved into the cell, and it will be surrounded or taken into a vesicle. It is called it to be as the phagosome, that is the body. Phagosome, that is the engulfed body within this. Here what will happen, these phagosomes are going to get combined with the lysosome. So here after the lysosome combination, it is going to become a combination of a complex of a phagolysosome. Here you can see phagolysosome, which is going to be like this. Now during this particular phenomena, what will happen? There will be the consumption of oxygen, that is this particular macrophages are going to consume the oxygen in more amount, that is the increase of the consumption of oxygen takes place, and the phenomena is called the respiratory burst. Now this process of respiratory burst produces reactive oxygen containing molecules that are antimicrobial. Now when you are going to consider the lysosome, when it is going to combine here, during this process, that is after formation of the phagolysosome, there will be the action of this particular oxygen, reactive oxygen containing molecules act on the pathogen. Because of that, the pathogen is going to be get killed. So here it is the antimicrobial activity by reactive oxygen containing molecules of the phagolysosome. The oxygen containing molecules are toxic to both the invader, that is the pathogen, and the cell itself. So they are kept in compartments inside the cell. This method of killing invading microbes by using the reactive oxygen containing molecules is referred as oxygen dependent intracellular killing, of which there are two types again. So that will be discussed. Now after this, what will happen? Whatever the pathogen has been killed, that will be removed by the macrophage cell, and it will be liberated into the external environment as the waste. Now the first type of this particular oxygen dependent production of a superoxide. Here also we have discussed that superoxide or hydrogen peroxide and other ROS will be produced during this particular accidental process of the interface of the bacterium and the activated phagocyte, and followed by this particular phenomena. This particular oxygen dependent intracellular killing is of two types. So that in that first type is the production of superoxide, which is an oxygen rich bacteria killing substance. The superoxide is converted to hydrogen peroxide and singlet oxygen by an enzyme called superoxide dismutase. Superoxide also react with the hydrogen peroxide to produce hydroxyl radicals, which assist in killing the invading microbes. Also can be in the presence of redox active metal ions. Guys, as per the explanation here, we can see that the superoxide is going to form the hydrogen peroxide and the singlet oxygen in the presence of the superoxide dismutase. So here you can see the superoxide is going to react, and there will be the formation of the hydrogen peroxide. Now here the same reaction has been depicted here. This is the one form of the reaction, and here again we have taken the similar reaction. So you can observe the reaction both here in the single equation and or here in the multiple equations. So here when you are going to consider, the superoxide is going to react or get converted in the presence of the superoxide dismutase to the hydrogen peroxide and the singlet oxygen. Here also you can see that the superoxide is going to give rise to the H2O2 plus singlet oxygen. Now when you are going to consider again, the superoxide also react with the hydrogen peroxide to produce hydroxyl radicals, which assist in the killing of the invading microbe. So here the superoxide has formed the hydroxyl radicals, and when you are going to consider the same further is that also can be in the presence of the redox active metal ions. Hydroxyl radical can be produced via the metal ion. Now when you are going to consider in this equation, we have seen the superoxide action here, and also here the production of the hydroxyl radicals. When you are going to consider here, the presence of the metal ion is going to be one of the reason for the formation of the hydroxyl radical. So here this is how the action of the enzymes metabolically, or the presence of the metal ion metabolically in the internal source, first type, that is the oxygenated dependent production of the superoxide, is going to takes place, and the hydroxyl radical is also going to takes place, and also the singlet oxygen is also going to takes place. So this is regarding the first type. And the second type is, in this it involves the use of enzyme myeloperoxidase from neutrophil granules. When granules fuse with a phagosome, myeloperoxidase is released into the phagolysosome, and this enzyme uses hydrogen peroxide and chlorine to create hypochlorite, a substance used in domestic bleach. Hypochlorite is extremely toxic to bacteria and also to the cells. Now here you can see this will be the neutrophil. Here within the neutrophil it is having the phagolysosome. The reaction which has been shown here, the same has been depicted here. The enzyme myeloperoxidase is going to be present in the form of the neutrophil granules, and it is going to be present in the phagosome. And after the formation of the phagolysosome, the enzyme uses hydrogen peroxide. So here this is the hydrogen peroxide, and it is going to take the chlorine to create the hypochlorite. Here this is the myeloperoxidase enzyme, and further a substance used is to domestic bleach. Domestic bleach in the sense here, after the killing of the microbes, that has to be cleaned from the cells. That is the domestic bleach, where this domestic bleach is the hypochlorite, which is extremely toxic. Here this is the hypochlorous, which is extremely toxic, and it is going to show its effect as the free radical. So here these are the two types of the oxygen dependent production of the free radical: the superoxide, hydrogen peroxide, hypochlorite, or any other form of the free radical. Now here this is the accidental type of the free radical production during the metabolism within the cells. Now when you are going to consider the free radical production in cells, where it is because of the deliberate reactions, is the here also we are discussing the same phenomena but in the different manner. Now when you are going to consider, there is also a large body evidence indicating that oxygen radicals are involved in intercellular and intracellular signaling. Now when you are going to consider intracellular or intracellular signaling, these both comes under the signal transduction phenomena, where it is going to play a major role in inducting a signal to perform a particular function within a cell by the other cells to a particular cell, or by the other factors within a particular targeted cell. That is what here the things are going to happen. Now when you are going to consider within this, the phagocyte NADPH oxidase FOX is a well characterized enzyme that generates high levels of superoxide and secondary oxidants in phagocytes as part of the microbicidal defense mechanisms by which these cells function in host defense. Now here it is as similar as here what we have discussed, but here the mechanism or the metabolic activity is different compared to this. So here what is happening, the enzyme phagocyte NADPH oxidase, which is shortly called as FOX, is going to characterize the generation of the high levels of superoxides and secondary oxidants, which will be responsible for the microbicidal defense, which are going to act as the defense, as the host defense, which is going to act as the host defense for the infections. Here you can see in the diagram that microbes, inflammatory mediators, which are going to act on the cell. So here whatever the microbicidal defense has been generated in the form of the superoxide and the secondary oxidants are going to act on this particular infections or the pathogens. Here what is happening is that the catalytic moiety of FOX is the membrane associated flavocytochrome GP91 FOX, which is inactive in resting phagocytes. Now here, when you are going to consider catalytic moiety, catalytic, you know that every enzyme is a catalyst, where it is going to act as the catalyst and it is going to complete the reaction. Moiety is nothing but, here it has two functions, that is it can rest the metabolic activity or it can activate the metabolic activity. That is the function. Either it can switch on or switch off the activity. That is the main feature of this FOX enzyme. So it has the catalytic moiety activity on the associated flavocytochrome GP91 FOX, which is the transmembrane protein, which is the receptor again, or it can be a signaling path. When you are going to consider which is inactive in resting phagocyte but becomes activated in the phagosomal membrane by assembly with regulatory subunits such as P47 FOX, P67 FOX and RAC. Here you can see P47, P67 and P40, and here it's the RAC, and here one more. A new family of homologues of GP91 FOX, the NADPH oxidase, dual oxidase. It can be NADPH oxidase NOx or dual oxidase family, which now contains 7 membrane. So here the complete set complex is the family. It will be having the 7 membrane, has recently been described, and these are expressed in various cell types. Now these are going to form the assembly when there is the microbial influence, where there will be the influence of the microbial. Because of that, what will happen, this assembly is going to form, and due to this, what will happen, this assembly is going to be initiated by the phagocyte NADPH oxidase FOX. Here you can see it is going to make the resting phagocyte to activated phagocyte. Because of this, what will happen, this is going to express the various cell types including the epithelium, smooth muscle cells and the endothelium. These enzymes deliberately generate superoxide and secondarily produce other reactive oxygen species including hydrogen peroxide. Now you can see here there is the action of the FOX enzyme, that is the phagocyte NADPH oxidase, which has influenced the formation of this particular complex, that is the 7 members of the family, which are the homologous in the sense these are the complex of the proteins which has been made, and here they are going to take part in the formation of the oxides, that is the radicals. That is, it can be the superoxide or the secondarily to produce the hydrogen peroxide. It is happening deliberately. Here it is happening deliberately, where they are generating this particular free radicals. So this is regarding this particular mechanism of the production of the free radicals deliberately on catalytic reaction basis. Now next is another major source of free radicals under normal circumstances is the electron leakage during the electron transport chains, that is electron transport chain in the mitochondria and endoplasmic reticulum to molecular oxygen generating superoxide. Here during the cytochrome C oxidase complex is highly efficient to reducing oxygen to water, and it releases very few partly reduced intermediates. However, small amounts of superoxide, anion and peroxide are produced by electron transport chain. Now here already we have spoken that the formation of the free radicals within the mitochondria, that is by the electron transport chain, is helpful in healing if there is the formation of these free radicals at the optimum required level. But whatever the process of the formation of the free radicals to the optimum level or to the excess level is going to remains the same. So that is what here we are trying to say. So the cytochrome C oxidase complex is highly efficient in reducing oxygen to water, and it releases very few partly reduced intermediates. Here reduced intermediates in the sense the whatever the reduced form of the products has been formed, that is called it is the reduced intermediates. However, small amounts of superoxides, anion and peroxide are produced by the electron transport chain. Particularly important is the reduction of coenzyme Q in complex 3 as a highly reactive UB semiquinone. UB semiquinone free radical is formed as an intermediate in the Q cycle. Now here cytochrome 3 coenzyme Q, here there will be the release of the superoxide, formation of the hydrogen peroxide, and metal ion effect, formation of the hydroxyl group, and there will be the reduction by the enzyme, formation of the water reduced, and it can also catalyzed to the water, that is hydrogen peroxide catalyzed to the water and the oxygen. Now UB semiquinone free radical is formed as a intermediate in the Q cycle. So this semiquinone or UB semiquinone is a free radical resulting from the removal of one hydrogen atom with its electron during the process of dehydrogenation of a hydroquinone, such as hydroquinone itself or catechol, to a quinone, or alternatively the addition of a single H atom to a quinone. It is highly unstable. Now here during this process a particular benzene ring kind of the hydroxyl has been formed, that is here they have mentioned a form of the catechol, hydroquinone itself or catechol in the sense that is the benzene ring kind of the hydroxyl, where the hydroxyl will be attached to the benzene ring, and due to that production of this particular quinone or alternative, it is going to add up the single H atom to form the quinone. So because of this it is going to become unstable. So here again it is the hydroxyl group formation. So this unstable UB semiquinone species can lead to electron leakage when electrons transfer directly to oxygen, forming superoxide. Now here during the metabolic process of the electron transport chain, there are certain byproducts formed. Those byproducts formed are literally unstable. So here what will happen, there will be the leakage of the electron and forming of the oxygen or into the superoxide, as we have discussed here. Well, coming to this slide, here we discuss the action of the production of proteins, in the sense protein folding process is going to take place in the endoplasmic reticulum, where certain redox communications is going to take place, where this redox communication is going to act as the mediators, where there will be the possible formation or production of the reactive oxygen species by possible surrounding sources. Now here the diagram which has been taken into consideration is showing the folding of the protein. Here you can see the protein folding is taking place, and this is ER. ER is nothing but the endoplasmic reticulum, and it is the cytosol where the ER is present. Now here there is a long process of the formation of the protein folding, during which there will be the protein folding process, and there will be the formation of the hydrogen peroxide and the superoxide, where this will be considered by the glutathione reductase. It is an enzyme which neutralizes the certain free radicals. Here also you can see with different sources there is the influx of the oxygen, and also here you can see there are certain toxin compounds which are going to form the ROS, which will be recycled in the sense here these ROSs will remain ROS but neutralized compounds, safe compounds. So here in this manner there will be the release of the free radicals during the electron transport chain process. So, and also here it is the protein processing or the protein folding processing within the endoplasmic reticulum. So here, coming to the next point, a major source of the free radicals that contribute to reperfusion injury, which we have studied earlier, two or three slides earlier, which has been again spoken here, where it is going to cause or remain as the major source of the free radicals that is being contributed through reperfusion injury in electron transport chain. The electron transport chain produces free radicals at very low rate under basal conditions in the normal conditions, but at elevated rates during conditions such as ischemia. I have spoken very briefly and very cleanly that ischemia and reperfusion is going to produce the high amount of the free radicals within the mitochondria. Reperfusion induced increase in the free radical production are often attributed to declines in the activities of electron transport chain complex upon ischemia that could in turn lead to an impaired overall rate of the oxidative phosphorylation. Here in the sense, when there is the ischemia reperfusion injury happening, during that time the stress or whatever the influence has been or the stimulus has been created on the mitochondria, it is going to lead to the impaired overall rate of oxidative phosphorylation. Oxidative phosphorylation is the one of the main metabolic process of the mitochondria, but here due to the ischemia reperfusion injury or reoxygenation of the blood flow is going to create such a stimulus or influence on the mitochondria of those cells within the blood flowing vessels is going to impair the oxidative phosphorylation. Because of that, it is going to lead those cells to a different form of the expression. It can be a major disease symptom. So this is regarding the free radical leakage during the electron transport chain in mitochondria and also the endoplasmic reticulum. In this slide, we will discuss the unusual circumstances which are other than the internal source of free radical production in cells. Those can be ionizing radiations, ultraviolet light, and other forms of high energy exposure. Free radicals are produced in cells in general by electron transfer reactions, which can be enzymatically mediated or non-enzymatically mediated. Now here, when we are consider electron transfer reaction is nothing but here the reaction is going to be taking place on the high energy basis, where it will be substituted or added by ionizing radiations of ultraviolet or X-ray or gamma rays. So here, because of this energy exposure, there will be the electron shift or transfer where it will be an electron transfer reaction. Either this electron transfer reaction can be aided or mediated by enzymes, or it can be non-enzymatic also. Now here within this diagram you can see that the DNA, the nuclear genetic material, is under the stress created by the deficit or difference in the electron, neutron or proton under the alpha helium 4, where it will be the direct impact on the DNA molecular structure, where this impact is going to take place by reacting these electrons of alpha helium 4 with the elements of the DNA. Now when you are going to consider the indirect form, here you can see that the X-ray or gamma rays are going to show the impact on the water molecules, where it generates the free radicals releasing the oxygen, which is again going to get converted to the superoxide and the peroxides. Now further, after this action, both the direct and indirect, it is going to lead to the formation of DNA damage. This is going, either it can push the DNA to its repair mechanism through polymerases, and if there is a failure in the DNA repair, it can proceed for the cell death. If it is the DNA damage, if it is non-repairable, again it is the cell death. If the DNA damage is not pushed to the DNA repair or non-repairable damage processes, where the necrosis or the cell death has to take place, where if it is not going to take place, if the cell replicates with the DNA, after the DNA replication, it is going to lead to the hazardous diseases, like many you know as such. Now here, this is regarding the unusual circumstances. The usual circumstances what we studied are internal metabolic processes which will be responsible for the release of the free radicals. So here, this is how the free radical production takes place in cells.